So, is this on? No. Let's find the button. Is it on now? No? Hello?
Yeah.
Okay. So, we studied hypoxia in the Maine waters.
I'm Sophie, I'm from New York City.
I'm Molly, I'm from Booth Bay, Maine.
I'm Mariah, and I'm from Alfred, Maine.
Okay. Okay, so from what you learned from acid, phytoplankton produce oxygen. So why would high phytoplankton abundance lead to low oxygen in the waters if they produce half the oxygen we breathe? This is a question that drove us through most of our research.
So now we'll answer it for you. Nitrogen levels in coastal waters are increasing dramatically due to human impact. The way we have contributed to this is through agriculture, sewage, and increased population along the coastline, which leads to nutrient-rich runoff represented by the red circle.
So the reason we chose to study nitrogen is because it's a limiting nutrient to the phytoplankton abundance. When there's an excess of nitrogen, there is a massive bloom of phytoplankton.
Shown by the blue circle of the algal blooms. Okay. When phytoplankton die, they sink to the bottom and they're broken down by bacteria. Bacteria consume all the oxygen that they've produced and create low oxygen areas, and also known as hypoxic zones or dead zones.
So, to recap, high nitrogen inputs leads to large algal blooms, which leads to oxygen depletion.
Because of hypoxia, fish and other organisms are forced to relocate because they cannot survive in a low oxygen environment. Dead zones are incredibly damaging to the aquatic ecosystems and to the fishing industries. This picture is of a massive fish kill.
Our research questions. We asked the questions, does hypoxia affect Maine coastal waters? And more specifically, what is the relationship between nitrogen, oxygen, and phytoplankton abundance in the Maine coastal waters? And how does the relationship change between depth and location?
So, our hypothesis is, if there is a larger abundance of phytoplankton at the surface and higher levels of nitrogen, then there will be lower levels of oxygen as the depth increases.
So we went to 4 different locations to sample for this project. We went to Freeport Town Dock, that's located on the Harrisicot River, Harso, Maine on Mackerel Cove, and we sampled 2 locations up at Maine Maritime in Penobscot Bay. We sampled at the mouth and farther up.
Okay, at Maine Maritime Academy, we use the CTD, which is shown in that top picture right there. A CTD is an instrument used to determine the conductivity, temperature, and depth of the ocean. At Freeport Town Dock and Mackerel Cove, we used a depth profiler, also known as the science source. We collected surface samples with a bucket, and we concentrated all our samples, capturing all phytoplankton larger than 20 microns, as Sophie's doing in that picture down there. And that's me and Mariah trying to figure out the science source.
In all of the samples we collected, we found 5 parts per million of nitrogen. This is an incredibly high level of nitrogen. It converts from 5 parts per million to about 4.99 milligrams per liter. In a study we found consisting of nitrification on the Maine coast, we saw that the nitrogen levels were drastically lower than what we found.
So this graph shows the phytoplankton abundance we found in each location. The y-axis is the phytoplankton abundance in cells per liter, and the x-axis is the locations we sampled from. As you can see, we found more phytoplankton at the depth of all of our samples.
This doesn't exactly support our hypothesis because we thought we would find more phytoplankton at the surface than at depth. However, all of the phytoplankton we found at depth were clearly clumped together and had sunk to the bottom, and they were in declining health. This suggests that we were observing the end of a pulse of the North Atlantic spring bloom. This picture is of a cell we saw most often, Copacods socialis. Because we saw many dying cells at depth, this led us to believe that we would find lower oxygen at depth as well.
This graph shows the relationship between oxygen levels and the depth of the sample. The x-axis is depth, which we represented as meters from the surface over total meters to normalize the differences between surface and depth at each location. The y-axis is dissolved oxygen in milligrams per liter. As you can see, in 3 of our locations, the Penobscot Bay location, the Harpswell location, and the Freeport location, the oxygen was lower at depth than at surface, which supports our hypothesis. Freeport Town Dock was where we observed the largest difference of dissolved oxygen levels between surface and depth.
So this graph is comparing temperature to oxygen at each location with temperature in Celsius on the y-axis. So the higher the bar is, the warmer the water was. And the oxygen levels in milligrams per liter on the x-axis. So the farther right the bar is, the higher the oxygen levels were. As you can see, the green line is Freeport, and it was the warmest water temperature, and bacteria rely on warm waters to actively break down phytoplankton, which helps explain why the oxygen levels in Freeport had the most contrast between surface and depth, which is also the trend that we saw in the previous graph. And Freeport is the most impacted by humans because of all the agricultural fields causing nitrogen in the runoff. And the other sites we sampled from were more remote.
Okay, this is my favorite slide. Hypoxia is not a major problem in Maine because most of the time waters are too cold for the bacteria to actively consume phytoplankton biomass. We saw high nitrogen levels and low phytoplankton abundance at depth, and high phytoplankton abundance at depth, and lower oxygen at depth. However, we did not see oxygen levels that raised concern for hypoxia in the ecosystems. But we saw that the warmer waters showed lower oxygen levels at depth. This means that as climate change progresses, the oceans become warmer, hypoxia will become a greater threat for the Maine coastal waters.
We would like to thank Carrie Whitaker for taking us to collect data, helping us through times where all 4 of us were confused, and overnight shipping the nitrogen tablets when the strips wouldn't work and the other tablets had expired.
And Laura Carson for also taking us on data collection, reading our papers countless times and giving us feedback, and for all of the beautiful pictures.
We would like to thank the people at MMA who helped us with the CTD, especially Carla, Xander, and Matt.
We would also like to thank our 12 other sisters for helping us with our data when it was frustrating and confusing. A special thanks to Kate for taking over the data collection at the second CTD location.
And Gail for all those lovely bagged lunches, for the days where we missed lunch for data collection, day after day.
And finally, we would like to thank all of you for coming.
Questions for the hypoxia group? Yes.
I'm just curious how confident you are about the accuracy of your nitrogen level measurements.
Question. Repeat the question.
So,
Repeat the question.
I was also alarmed. Our oxygen levels, nitrogen levels, how confident we are in the accuracy. So because it's so early in the winter-spring bloom, we would expect nitrogen levels to be higher. I'm still not very confident that 5 is up there, is like how drastic it was. And we also didn't have like a control sample to see if the nitrogen tablets were actually working effectively. We just kind of trusted them. They were brand new, so.
Other questions? Yes.
When you see like alewives or a big fish kill wash up, is that hypoxia or that could be a whole bunch of different things? But is that one of the causes in Maine, or are you thinking that Maine doesn't get?
Repeat the question.
So when you see a lot of fish that are dead, is that only because of hypoxia, or could it be a lot of different things? So sometimes when fish like all swim into a harbor, since there's so much population of them, they can consume all of the oxygen in the harbor and like not be able to turn around and get back out to the oxygen-rich open ocean. I guess oxygen low levels in harbors could be a reason to speed up this process, but I feel like they would consume all the oxygen in the harbor themselves.
Any other questions? Okay, well, let's give them another round of applause.